This is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be discussing about gibberellin. Gibberellin is one of the promoter, right? And along with the auxin, it also leads to growth, or it participate in growth, but primarily it is known for the growth of internodes, right? So gibberellin occurs in both flowering as well as non-flowering plants, okay? And there has been as many as 126 gibberellins has been identified, but out of them what we say that GA3 is the most common one, okay? And all the forms of gibberellins are actually derived from what we call gibbane ring, as you can see in the diagram. So this is what we call it is a gibbane ring. So all the 126 GA is going to have the same structure of the gibbane ring, right? Let's talk about a little bit about the history of the gibberellic acid. Now, how this gibberellic acid came into existence, or how actually we came to know about the gibberellic acid. Actually, the credit goes to the Japanese scientist, and his name was E. Kurosawa. What he was doing, he was studying the seedling fullest disease of rice, okay? So this disease is caused by Fusarium moniliforme, that is the name of the perfect stage. But when he was working at that time, the perfect stage was not known, and at that time the fungi was named as a Gibberella fujikuri, and by that it belongs to ascomycetes. So later it was isolated and crystallized by Yabuta and Sumiki, okay, but the name gibberellin was coined by Yabuta. So credit for coining the name gibberellin goes to Yabuta, okay. So let's talk about biosynthesis. You see, gibberellins are the diterpenes, okay, and they are synthesized by acetyl coenzyme A via mevalonic acid pathway, okay, and it is present in both free and bounded form. There are three plant organs where the concentration of gibberellins are relatively high, and they are shoot buds, root tips, and developing seeds. Even among these three, the highest concentration is found in the developing seeds and the young leaves, right? Let's talk about the bioassay. So one of the most common bioassay for gibberellic acid is lettuce hypocotyl elongation test and serial endosperm test, okay? The movement of gibberellin is going to occur by all three methods, what we call simple diffusion, phloem transport, and xylem transport. So the direction of movement can be both upward as well as downward, right? Let's talk about the function. As I told you, the main function of gibberellin is the elongation of internodes, okay. So what happens? GA stimulates internode elongation and expansion of leaf, but mind it doesn't have any effect on root, okay. There is one more important function of gibberellic acid, and that is bolting. Let's try to understand what exactly is a bolting. You know, there are many plants in which there is going to have a very profuse growth of leaf, but internodes are shortened. Take an example of cabbage. You might have seen that in case of cabbage, there is a large leafy structure, but the internodes are suppressed. But if we use gibberellin in those plants, what happens? There is an elongation of the internodes, okay, and when the internodes get elongated, this particular phenomena is actually called as a bolting. So that is one of the important character for GA, okay. Have a look at few more functions. So what happens? GA promotes germination of seeds, especially in cereals. At the same time, GA also promotes flowering in long day plants, might it, but inhibits flowering in short day plants. Many a time this particular question is asked in NEET. Promotes flowering in long day plants, but inhibits in short day plants. GA is the only hormone that induces maleness, because the other hormones like auxin, cytokinin, or ethylene, they promotes femaleness, but GA is the only, we can say it is a male hormone. Now it helps in the development of parthenocarpic fruit. In the session of auxin also we have discussed that auxin, gibberellin, and ethylene, all the three are known for producing the parthenocarpic fruit, but out of these three, ethylene is the most potent one, right? And GA can substitute vernalization. I am sure all you know vernalization, that is the cold treatment to induce the growth, okay, So when those plants which require cold treatment, instead of cold treatment, if I give GA, then even in that case, they are going to perform. Okay, so that's why we can say that GA can replace the bundle action. Moreover, GA is also known for breaking the dormancy. There are different kind of dormancy. Okay, so primarily GA is known for breaking the dormancy. That is the most important function of GA. Okay, so that's all as far as the entire function of gibberellic acid is concerned, right? Thank you.